Welcome to my lecture. I am Dr.M.Isai from Department of Zoology. This video will explain the stem cell and regenerative medicine. It's a modern medical science. Before going into detail, let me give a brief introduction about the stem cell. Stem cells are defined as precursor cells. These cells has the ability to self-renew, continuously divide, and differentiate into any type of tissues or cells. These cells have the capacity or ability to branch out, change, and differentiate into two or more type of cells, and they are functionally differentiated one. This picture clearly explain about the definition of stem cell. Here, the bone marrow stem cells contain endothelial progenitor cells, mesenchymal stem cells, and hematopoietic stem cells. This endothelial progenitor cells can differentiate into capillaries and heart tissue. The mesenchymal stem cells can differentiate into heart tissue, skeletal muscle, connective tissue, adipose tissue, bone cartilage, liver, and neuronal cells. In the case of hematopoietic stem cells, the name implies that these can differentiate into red blood cells and white blood cells. The stem cells have the specialized features. They are unspecialized cells, that is blank cells. They're capable of dividing and self-renewing themselves for a longer period of time. And they have the potential to give rise to special cell types. What are the properties of stem cells? Stem cells have 5 different properties. Number 1, totipotent. Number 2, pluripotent. Number 3, multipotent. Number 4, oligopotent. Number 5, unipotent stem cells. The totipotent stem cells are derived from 1 to 3 days old embryo. These cells can develop into a new individual. And the pluripotent stem cells are obtained from inner cell mass of blastocyst, that is from 5 to 14 days old embryo. These cells can form any cell types. In the case of multipotent stem cells are from fetal tissue, cord blood, and adult stem cell. These cells differentiate, but it can form a number of other tissues. In the case of oligopotent, these cells are from lymphoid or myeloid stem cells. These cells can differentiate into only a few cells. And the last one is unipotent stem cells. These cells are from adult muscle stem cells. These cells produce only one cell type of their own. For example, hepatoblast cells can differentiate only into hepatocytes. Coming to different types of stem cells. In general, stem cells are broadly classified into 2 different types. One is adult stem cells. Second one is embryonic stem cells. And the adult stem cells are derived from various tissues in the human body that can branch out into many different types of cells. Hence, the cell is called multipotent stem cells. The next one is embryonic stem cell. It's also referred to as ES cells. These cells are derived from pre-implantation embryos. They are unique because they are pluripotent stem cells. It is true because they can develop into all cells and tissues, and they self-renew indefinitely in their undifferentiated state. This picture explains about the sources of stem cell and its potency. Single cell embryo contain totipotent stem cell. Pluripotent stem cells can obtained from 5 to 7 days old embryo. These cells are embryonic stem cells. And the multipotent stem cells are obtained from infant. These cells are from from infant cells or cord blood stem cell and placental stem cell. And the multipotent stem cells also obtained from infant and adult. It is called as adult stem cells. This table explains about the embryonic and adult stem cell comparison. The embryonic stem cells are totipotent or pluripotent stem cells. It is very easy to isolate and identify. We can harvest large number of cells from inner cell mass of blastocyst or embryo. It may cause immune rejection. When we are using embryonic stem cell, we need to face ethical issues. Coming to adult stem cell, these cells are multipotent. It is very hard to identify and harvest. We can get only a limited number of cells at the time of harvesting. These cells cause immune rejection very less. And there is no ethical issues when we are using adult stem cells for our experiment. This slide explains the stem cell line. The stem cell line take through ups and downs of stem cell roller coaster. In the year 1981, Martin Evans at the University of Cambridge is first identified the embryonic stem cell in mice. In 1997, Dolly the sheep was produced by Ian Wilmot and colleagues at the Roslin Institute, Edinburgh. In this process, they fused sheep egg with udder cells and implanted the resulting hybrid into surrogate mother sheep. Researchers speculate that the similar hybrid process applied in stem cell research. Here, they fused embryonic stem cell with adult stem cell from particular patient or particular person that could be used to treat disease or injury. In 1998, James Thomson and John Hihart isolated human embryonic stem cells and they cultured in the laboratory. In August 2001, President Bush limited federal fund of research on human embryonic stem cell line because in this process, human embryos were destroyed. But Bush does allow to continue the research on human embryonic stem cell line that were created before the restriction were announced. In the year 2006, adult cells were reprogrammed into induced pluripotent stem cell by Shinya Yamanaka of Kyoto University for the invention of induced pluripotent stem cells from matured cells. He was awarded Nobel Prize in the year 2012. In 2009, President Obama lifted the 2001 restriction on federal fund for the use of human embryonic stem cells. Hence, in the year 2010, human embryonic stem cells were used to treat the person with spinal injury. Similarly, 2012, human embryonic stem cell shows medical promise in the treatment that is blindness. In 2014, world's first trial of therapy based induced pluripotent stem cell to treat the form of age-related blindness. Then, what to do with these stem cells? If you have a sample like peripheral blood or bone marrow, first we have to isolate the specific cell types For example, if you are selecting CD34 or CD38, we have to choose a specific or appropriate marker for the isolation of stem cell population. For the isolation, we have to use magnetic activated cell sorting system, that is MAX. After isolated cell, the cells were characterized with the help of additional marker in fluorescent activated cell sorting system, that is FACS. After characterization, we need to expand these cells for the differentiation study. At the time of expansion, we need to keep the cells in a controlled condition, and also it should be free from mutation, and to sufficient quantity we have to culture for the differentiation. Coming to regenerative medicine, it is the emerging field of therapy. It is also known as cell therapy. It is referred to as the treatment that are funded on the concept of producing new cells to replace the malfunctioning or damaged cells as a vehicle to treat the disease or injury. Our focus is mainly on the development of effective methods to generate replacement cells from the stem cells. This is especially true of diseases associated with aging, such as rheumatoid arthritis, arthroarthritis, Alzheimer's, Parkinson's, type 2 diabetes, heart failure, and aging of immune system known as immunosenescence. It is true because of replacing damaged or malfunctioning cells with fully functional one may be a useful therapeutic strategy in treating many of these diseases and conditions. This picture explains the cell therapy. Here, the peripheral blood, from the peripheral blood, we have to isolate the stem cell. The isolated stem cells were manipulated, expanded, and reprogrammed. These reprogrammed stem cells were transplanted into patients. Why opt for regeneration? Mainly two reasons. One for shortage of donor, and the second one is transplantation rejection. These unlimited supply of autologous cells, like induced pluripotent stem cells, could be used to generate transplant without any immune rejection. This picture explains about the organ regeneration from the patient's bone tissue. Here, the bladder cells were isolated and cultured in a controlled condition. After several days, these cells were seated on the scaffold and they were cultured for 7 weeks. After 7 weeks, the scaffold were attached to bladder, into which it integrated. What are the potential uses of stem cells? In basic research, the clarification of complex events that occur during human development and understanding molecular mechanism of or molecular basis of cancer, such as molecular mechanism of gene control, differentiation of stem cell, and stem cell theory of cancer. In the field of biotechnology, stem cells can provide specific cell types to test new drugs. For example, the availability of pluripotent stem cell would allow drug testing in a wider range of cell types to reduce the animal model. In cell-based therapies, regenerative therapy used to treat the various kinds of diseases and disorders such as Parkinson's disease, Alzheimer's disease, heart disease, type 2 diabetes, severe burn, stroke, rheumatoid arthritis, and arthroarthritis. In the case of gene therapy, stem cell act as a vehicle after they have been genetically manipulated. I would like to give an example for regenerative medicine. In a heart failure patient, doctors are able to improve the heart function and reverse tissue damage. For this, they used stem cells from patient's own heart tissue. In this picture, progenitor stem cells were obtained from patient's own heart tissue. These cells were isolated and cultured along with the feeder layer. These cells, the stem cells, can differentiate into heart muscle cells and vascular cells with the help of carcinogenic factor. These cells were transplanted back into patient's body. Hence, I concluded that stem cell and regenerative medicine is still in its infancy. Very particularly, human embryonic stem cell research is a decade old, but adult stem cell research has 30 years head start. This field holds hope for curing or improving treatment for more than 70 diseases, and also veterinary applications needs to be studied. Thank you for listening.